when I arrived, actually when I was a postdoc at Caltech, the Human Connectome Project had been funded. It got funded in 2010, and they did their first data release, I think in 2011 or 20. Yeah, I was there when they did their first data release. And we got a whole, I think they mailed out this hard drive and you can pull their data. And it was pretty exciting, I think, for what they were trying to do, both with regard to pushing the limits on the data quality, imaging data, but also the fact that the whole purpose was to share the data with the scientific community. And when I came to interview at the University of Minnesota, I met with Camille Ugerbil, who's the director of the Center for Magnetic Resonance Research and the co-PI with David Van Essen of the Human Connectome Project. And I asked him directly, what's next? And fortunately for me, and I think for the University of Minnesota, he was privy to some of that information. He said, I think that they're going to push for a lifespan call, and I think that the next steps are disease connectomes, and they're going to be pushing for non-human primate connectomes projects, collaborative efforts. And so I was able to kind of get going and launch with the expectation that there would be a call for connectome projects across the lifespan. And certainly clearly there were. These first two came out first, one on what they call development. It was they were calling for 5 to 21-year-olds. And then they made a call for the aging cohort, which was, I think, 50 and above. And shortly thereafter, and these were both big projects, kind of like, I think they were 4 years, around 15 or 16 million per project. And then they released the call for the baby connectome. And it was kind of like an afterthought, maybe not, but it was a much smaller scale. And so we actually, for each of the other two projects, can you guys see if I use my cursor? Does that, yeah, okay. So each of these two projects, there are 4 sites that include the University of Minnesota, Washington University in St. Louis, Harvard, and UCLA as the data collection sites. And there just simply wasn't the money to go for that many sites for the baby connectome, and so it ended up being University of Minnesota, and we ended up joining forces with the University of North Carolina at Chapel Hill. As Lynn mentioned, that's where I did my training, and so you can reference these papers. They give you a pretty good rundown of the objectives, what we're trying to do, and the imaging parameters are all there, the sequences, and the behavioral battery, all that good stuff. So the specific aims were to obtain HCP compatible MR images and behavioral assessments. So one of the objectives was to try to unify across these projects. So with regard to thinking about what imaging parameters are you going to implement. They wanted to be able to collapse the data eventually. And so that, in some regards, I want to say, I shouldn't say handcuffed us, but it kind of did, because there may be some choices that you would make with an infant protocol that don't necessarily correspond or that aren't optimal as compared to an adolescent or adult cohort. But at any rate, we think that what we were doing, while maybe not optimal, was still way ahead of the game. And I think with regard to kind of precision of what we were doing. So the next aim was to provide effective, robust quality assurance, quality control, to integrate novel and dedicated early brain development image analysis tools into the existing HCP pipelines. That's still kind of in progress and being overseen by the group at UNC. And then lastly, to share the data with the scientific community. And I had really, really hoped to be able to make kind of like a first announcement that the data were on this presentation, that they're available. They're all at they're not all there, but about 3/4 of the data are at the National Data Archive, and I don't know what they're waiting for. I'm gonna keep pushing them over the next couple of weeks to release it, to identify the release date, which I hope will be in August. And then the final data set will come in like December or something like that, everything that has been collected. So what are we doing here with these? So those are the aims, but the objectives really are to create an opportunity to characterize development of the normative connectomic architecture across the first 5 years of life, and to create an opportunity to map key behavioral domains to underlying neural connectomic architecture, and then to create an opportunity to compare to high risk or atypically developing samples. I say to create an opportunity because there weren't scientific aims in the Baby Connectome Project. It's really about collecting, it's about the design of the study, who's going to be able to deliver, and are you going to be able to deliver high quality images? And so that said, I mean, I've been, we didn't have time for like writing up papers and things like that. I've been very fortunate in that I've had great graduate students who've been able to leverage this award to kind of get awards for themselves to work up the data. But to give you an idea, to go back kind of this first objective, create an opportunity to characterize development. I think what set us apart in the review was our design, and we implemented this accelerated longitudinal design. And the next slide will kind of show you that better. But we created these cohorts so that we are covering kind of the birth to 60-month period more densely in the first 24 months of life, because that's when there's more dynamic change and more within person variability than between person variability that you have to deal with. And what's unique is the density of sampling in our longitudinal cohorts. So in our youngest babies, we were trying to get 6 visits. The last one was just the behavioral visit, but essentially five imaging time points. And then in this cohort B, where there's like 60 kids, we're trying to get 6 imaging time points, and so on and so forth. And this is a way to visualize it. The figure is old, but the numbers are pretty accurate. So it's around 447 children that we're going to deliver to the scientific community. Around 960 scans, where about 100 of these kids are cross-sectional. So the vast majority of the kids are going to have 2 to 4 scans. But this figure is old, but this gives you an idea of what the accelerated longitudinal design will look like, with kind of each dot being a scan, and each row being a kid. So we're getting 0.8 resolution T1s and T2s to derive some of these metrics that you would expect: cortical thickness, surface area, cortical complexity, Sokal and gyrification index, and to do these kind of novel parcellations. We're also getting DTI. I can say that the DTI is it was a little tricky because I didn't mention this at the outset, but essentially I had an R01. I got an R01 in 2014, in which I built the imaging parameters around what I anticipated the lifespan connectome project, the developmental connectome project, would implement, with the hope of being compatible, right? And the idea being that when the baby connectome RFA is made, I'm going to be the only one in the world that has data already that will be compatible with what's already being proposed. Now, what we proposed was a diffusion imaging sequence that was a 2-shell sequence that if we got through all 4 runs, it was going to be around 20 minutes and around 180 volumes or something like that. We decided as a group, as a consortium, to change the diffusion sequence at the onset of the baby connectome. So I had collected a bunch of data that was going to become part of the baby connectome with the different diffusion sequences. It was these different B values and different number of volumes. But at the onset of the baby connectome project, we changed to this 6-shell sequence. So there's going to be slightly less kind of total diffusion data, unless you can extract certain, which you could do, you can extract certain parameters from both sequences. The resting state, and there's just a nice little cartoon of everybody's favorite on here, the corpus callosum. We're also getting resting state data. Here we were pushing the limits, and when we were pretty consistent with the The other connectome projects, all of the connectome projects implemented the same resting state sequence, and we're pushing the limits with regard to the resolution and the acceleration. This is a 2.0 isotropic resolution and a multi-band factor of 8. I think it's good data. I think it's really good data. If I had it to do over, I may not have kind of taken my foot off the accelerator to some degree, but I think it's going to be really good data. Okay, and then a series of cognitive behavioral assessments with the idea. This was kind of one thing that I took pretty seriously in thinking about. I didn't want to just do an imaging study, and we have some good phenotypic characterizations. There's some that are just kind of like run of the mill. What are you using to characterize developmental level? We're using the Mullen. It's pretty good. It's a pretty good measure up until around 36, so above 36, it's kind of, you may want to implement something else, but for consistency's sake, we used it. The other two that I would highlight here is we have a good measure of executive function that begins, we started at 24 months, but we think at 30 months and later, it works really well. And then this is called the Dimensional Joint Attention Assessment. This is an assessment that I developed as a graduate student. I'll show you some results from that in a minute. So there is a kind of psychological model behind the design as well, and the implementation of the phenotypic battery. So all of the data is stored in Loris. I don't think I need to spend too much time here, other than to say that we feel good about how the data is being managed, and while Greg Farber and the National Data Archive are going to be discouraging us from sharing data, they want people to go to the National Data Archive to pull data. I kind of operate under the assumption that I don't want any barriers to sharing data. And so we're managing data in a way that we can share directly with people with certain kind of agreements in place. So yeah, I include this just because this is a lesson learned from this project. To do this kind of data management requires people and support. And so in thinking toward the future, we'll never not write in enough support for data management. Okay, so a glimpse at some data. We start with the behavioral data because I think that's one thing that also kind of set our project apart at the submission phase. And what we were able to do is essentially take this measurement assessment technique that I developed as a graduate student and collect a lot of normative data. And what you're seeing are the blue dots are normative data collected primarily in my lab, but there's about a handful of them, about 165 of the kids come from my lab, and they all have 2 or 3 data points. And then 40 come from our IBIS collaborators around the country. And then the green and the red are both groups of high-risk kids. The red is high-risk kids who go on to develop ASD, and the green are high-risk kids who do not develop ASD at 24 months. And so this is a really exciting finding. This is currently in revision. We're showing for the 3rd time now, there are only 2 other instances of this, of a overt behavioral marker that differentiates kids who go on to develop autism as a group under 12 months of age. So there are features that differentiate kids, but these are all things that you can't observe. So there's either eye tracking or imaging or EEG. So this is the first kind of direct overt marker. And then what was interesting to us is this finding, such that the high-risk kids who go on to who do not go on to develop autism are showing this kind of precocious ability at 9 to 10 months, such that they are significantly increased compared to the low-risk kids. And so this is catalyzing the story around promotive or protective factors potentially, which is kind of rare in the autism world.
Jed, what do you make of the finding at 17 to 18 months in the high-risk negative?
So they're just stabilizing. I think at 17 to 18 is the measure was designed to really characterize variability in the emergence of responding to joint attention. We think that you can only go up so high. So they're not different. These groups aren't different from each other at 15 months or 16 months. if you look cross-sectionally. But the slope is different because they started so much higher. But everybody, you know, if we had a bunch of data at 18 months, and I actually just collected a bunch of data at 18 months, kind of wasn't anticipating to, but the measure was really designed to be implemented from like 8 to 14. But by, you know, most kids will be able to do this. And so one question we're interested in is when there's a delay, how does that have cascading effects on subsequent social cognitive development? So the next slide is one that I wanted to, if we could just stop recording for a second, Lynn, folks, since I didn't want to necessarily share that one slide, but so leveraging the BCP, it's been a lot of work. We're, you know, at the scanner every night waiting for babies to fall asleep. And, but I think in the long run, we've been able to leverage it in interesting and exciting ways. But I also think it's going to be a great resource for the scientific community. And so in my shop, we've leveraged it for these 5 NSF awards for graduate students, which is fantastic because they can come in and they've been able to like work up the data, but also add on interesting questions that they have. We leveraged this in the grant with Lynn because a number of the parent report measures are similar to what we're going to be getting for the AgCC kids. And then I also leveraged it with this new R01 that I'll get to hopefully at the end. I'm not going very fast. We're talking about this new approach to population-based screening. So just a couple of slides on agenesis. The first is I wanted just to kind of be open and transparent and show you what Lynn and I are doing, because these data are going to be available, and I think that it's going to be a nice resource. But essentially, we wanted to do as well as we could describing behavioral development across the first 3 years of life. So we enroll infants at either 6 months or 12 months, and we get some data on temperament. We get some data on early emerging kind of autistic symptoms. We get something on sleep, and we get, this is the early motor questionnaire. This has been validated against the Mullen, so we're getting some kind of developmental motor milestones. We get a little bit of information from the parents. And then we at 12 months, you know, there may be some aspects that are continuous, as you can see, but then we're able to start getting some measures on early emerging atypical or maladaptive behaviors. That's the ITSE. It's validated for 12 to 30 months, and it captures aspects of internalizing, externalizing, kind of dysregulated behaviors, so on. This is a measure that I have been working to kind of validate. It's a measure developed by one of my colleagues. It's called the Repetitive Behavior Scales for Early Childhood. It's designed to capture a number of different topographies of repetitive behavior. This is language, CBCL, many of you are familiar with. Again, kind of broad-based early emerging psychopathology. This is another measure that we're very interested in, the VR-RSB. It's capturing aspects of reciprocal social behavior. It's kind of analogous to the social responsiveness scale, if some of you are familiar with that. It's the SRS. We're very interested in this because the SRS essentially, while it's designed to quantify continuously autistic traits that are continuously distributed in the population, it seems to differentiate, of course, kids with autism from controls, but kids with ADHD from controls, kids who are at clinical risk for psychosis from controls, kids with disruptive behavior disorders from controls. It seems to capture some aspect of social behavior that is abnormal in kind of a transdiagnostic fashion. The goal was to recruit about 150 kids and bring about 75 of them to Minnesota for a direct assessment between 24 and 30 months. So we're hoping that, you know, we can do that eventually, but we'll see. So the preliminary data that kind of informed this study came from a pilot that Lynn and I conducted, and where we're getting some of these measures 6, 12, 18, 24, and some cases older. But it was a kind of a bare bones battery and trying to do it on the cheap. But we now have some results that we're writing up and about ready to submit. And I'm going to showcase a couple of those findings, kind of some of the key findings. So this is data from the MCDI, the MacArthur-Bates Communicative Development Inventories. And so 2 key measures there are words produced. You're just asking a parent to report how many words does your child say. And then another is about gestures, communicative gestures. And here what's interesting is that this was one of few measures that we actually show a difference between about 57 kids with isolated AgCC. Some 10 of those kids have partial and the remaining have complete. And this eLab sample is my group, my sample is about 100 typically developing kids who all have longitudinal data. That's what we're representing here. So there are some subtle differences in words produced, parent report of words produced, and then parent report of gestures used by these kids. And we think this is notable because it's also going to be consistent with in every measure that we looked at, the only differences that we saw were in aspects of social behavior and social communication. So another kind of representation of some of these data here, I'm showing you some of the kids that aren't in the primary analyses, but kids with AgCC plus additional anatomical abnormalities. They're represented in the blue. The kids with isolated AgC are in the red, and then typically developing kids are in black. And so this is the measure that we're representing here is called the ITSE, the Infant Toddler Social Emotional Assessment. And so you get this nice, these internalized and externalized have been validated against the CBCL to give you an idea. Not showing any differences there. We are seeing differences in this competence, and this is social competence. So it's a measure that's very similar to kind of reciprocal social behavior. And this is where we see a difference. And so it's kind of, we also looked across this measure I was telling you about, the repetitive behavior scales, abnormal motor behavior, or kind of rigid adherence to routines or ritualized play. And there were no differences whatsoever. In fact, there was a significant difference in one of the scales, but it's in that the kids with AgCC were showing fewer than the typically developing kids. So the story that's coming out from our preliminary data is that there seems to be something about social communication that warrants further inspection and interrogation. And I'm presenting, I have put this slide in because I'm also going to be talking in more depth about these data in next week, 2 weeks, July 21st. And I belong to this consortium called the Baby SIBS Research Consortium. And it started with groups who were only studying high-risk infants who are at high risk for developing autism because they had an older sibling with autism. But there have been a number of groups now, including Jane Roberts, who's depositing data with a Fragile X. Megan Miller is interested in risk for ADHD, but she's studying younger siblings of kids with ADHD. So that's going to be an interesting kind of addition to the consortium. Emily Jones is studying kids with NF1, neurofibromatosis. And there's one other group studying kids with tuberous sclerosis that's also depositing data. So there's going to be this really kind of interesting constellation of infants at risk in this consortium repository with common data elements. And so I wanted to highlight that just because there will be multiple ways to go get these data, but these data are going to be shared with the BSRC as well. So IBIS, just to transition, this is a project that IBIS stands for the Infant Brain Imaging Study Network. This is I was a graduate student at the University of North Carolina when the study launched. It was designed to characterize brain and behavior development from 6 to 24 months in high-risk infants who are deemed at high risk because they have an older sibling with the disorder. There's this interesting phenomenon that we're trying to test, the timing and onset of this brain overgrowth hypothesis. And I'm not going to highlight those data. You can find those data. It's a very interesting Nature paper in which we describe the timing of brain overgrowth such that there aren't differences at 6 months, but there seems to be expansion of surface area between 6 and 12 months that leads to increased total brain volume at 24 months of age. But But we've just got funded last year to essentially replicate some of that work, mainly replicate work around prediction, individual level prediction based on MRI at 6 and 12 months of age, in which we were showing that we could predict at an individual level who would go on to develop autism at 24 months with greater than 80% accuracy. And what that does is it opens up opportunities for potentially for pre-symptomatic intervention if we can leverage these brain-based biomarkers. So Minnesota is a data collection site now, but these other 4 sites were the kind of original 4. And so we're on pause right now, but we're trying to continue to collect data and recruit infant siblings into a remote version. But we're very excited about that. But to date, before kind of this new instantiation, there's a cohort of about 600 high-risk infants and 200 low-risk controls. There's another extension of the project that's following the kids into school age at 8 to 10 years of age. But yeah, it's a pretty unique group. So I just wanted to highlight a few of papers of findings from our work, specifically on the corpus callosum and functional connectivity as it relates to this group. So corpus callosum, a couple of papers to check out, both kind of led by my close collaborator and friend, Jason Wolf. One in which he's showing that essentially the thickness of the corpus callosum was differentiating high-risk infants who want to develop autism from controls as early as 6 months of age. And that the effect sizes actually get smaller. So we're really interested in this idea of callosal structure and function being temporally limited and kind of functioning in a unique way during sensitive periods during development. This is another interesting study, just kind of showcase the breadth of the network. We're also showcasing that white matter development differentiates infants with Fragile X syndrome from controls, including aspects of the corpus callosum. We're also contributing to differences there. This next slide, I'm highlighting 3 papers that all point to this very interesting kind of hypothesis that I introduced you to. But this idea that the corpus callosum is doing kind of some heavy lifting very early in development, particularly the posterior projections of the corpus callosum that seem to be doing some temporally constrained type of integration of information early in development. And these 3 papers all show that slower development of the corpus callosum portends more advantageous outcomes, and which we think of as kind of this, supporting this less is more hypothesis, that there's some things that it's not advantageous to be precocious or to go fast. And I think setting up this region, specifically the posterior sector of the corpus callosum, the splenium, is projecting to aspects of the posterior hub of the default mode network, which is very well positioned to be a very important player in subsequent cortical specialization.
So why do you think that precocious development of the splenium is interfering?
So the idea would be that in order for kind of appropriate kind of tagging of salient information that might be, we don't know exactly what the posterior hub of the default mode network is doing, right? So, but what we can be pretty sure of is it's not what our, it's probably not doing self-reflection in babies, right? That people who study adults would suggest. It's probably doing something more along the lines of attentional selection. And there's nice work from Michael Platt and Ben Hayden doing single unit recording from PCC regions, showing that it's very important, kind of the baseline level of firing predicts with a high degree of accuracy kind of response times and reaction times. So it's kind of, we think of it as being critical for attentional selection, which is going to shape specialization with certain types of information. But there may be some advantage to not specializing too fast. We may need to determine, taking time to actually determine what are the most salient aspects of auditory information that's coming in, visual information that's coming in. And so there may be some advantage, at least in typically developing kids, to that this kind of slow pace of development in the posterior sector is advantageous. Next is I wanted to highlight kind of how we're thinking about brain and behavior with this gets back from moving from the corpus callosum to functional connectivity. This is the paper that I would point people to for this was kind of our first introduction to this analytic strategy. Essentially what we're trying to do is move beyond the lesion model into a model where we can actually examine how multiple brain networks communicate together to produce complex behavior. And so there have been a couple of two additional papers that come out on this method looking at different behaviors. But this work's led by John Pruitt and Adam Egebrecht is a PhD in physics turned fabulous neuroscientist. And so is kind of the brains behind this. But essentially you take a matrix, you have 230, say, functionally defined ROIs, and you generate a matrix of 26,000 ROI pairs, and you can transform that correlation coefficient associated. So you transform those correlations into a z-score, and you look at the association between that metric and your behavior of interest. And what that does is it generates this kind of, you can look for clusters of strong or weak correlations. And then the kicker is that you're testing whether that cluster is above and beyond some threshold that you would expect, and whether there's enrichment of significant connections or not. And that allows you to identify certain regions that are communicating with other regions, associated with other regions that are associated with the behavior. And then if you have developmental data, you can take the difference and look for changes across age. And that's one thing that you can do here. I'm showcasing essentially regions that differentiated the association between joint attention at 12 months and 24 months. And it appears that it's really these posterior regions are doing the heavy lifting at 12 months of age, and it expands to an anterior network, includes an anterior network at 24. So this enrichment approach is allowing us to look at more complex associations between networks, connectivity networks, and behavior. So the last little section that I wanted to introduce is kind of how we're thinking about translating what we're learning in high-risk samples for work in the general population. So this is a little figure that kind of a cartoon that tells you a little bit about the approach. This was our essentially the preliminary data that laid the foundation for the R01. We made a couple of changes with the R01 and how we're doing this, but I can talk about that as needed. But essentially, the idea is that for high throughput, population-based screening, we need something that works with parents that they'll do and that will work. And most often the case in the specific domain that I'm talking about and that I'm coming from the autism world. Many of you may know that the kind of best practice right now is a 20-item yes/no checklist that a pediatrician might give to a parent of an 18 to 30-month-old. It's called the MCHAT, the Modified Checklist for Autism in Toddlers. And it simply doesn't work. I used to say that it doesn't work well, but now I just leave it as it simply doesn't work. There's new data out from CHOP, from University of Pennsylvania, really highlighting that it's not working as the authors intended. And so we need to do better. And so one thought that I had, this was kind of fortuitous, but I started doing some work to validate these measures. When I've been talking about the RBS-EC, the Repetitive Behavior Scales for Early Childhood. And initials are the Video Referenced Rating for Reciprocal Social Behaviors, capturing aspects of social responsiveness, reciprocal social behavior. These were two new measures. This is a MCDI, again, the communication inventories. This is kind of a workhorse measure. We didn't feel like we needed to do work here. But I wanted to do some work just validating these measures. And I had an RA who said, well, why don't I put them in Qualtrics and we can get a lot of data on these measures. And so my department runs a participant registry with like 65,000 names, which is pretty unique for a developmental unit. But we were able to tap into a big sample, and I thought to myself, well, we should actually do more than just get data to validate. And we started brainstorming and thinking about what we could do, and that led to this approach, essentially, that we call phenoscreening. But essentially, what we're doing is we're incentivizing parents to give us more information than they normally would. Okay, we're paying them to tell us more about their child's development across multiple different dimensions of behavior. So we don't want just questions about what we think autism is in an 18-month-old. We want to be able to capture multiple dimensions of behavior. And so we get all of this information from a big sample. We implement some data-driven sampling techniques, stratification techniques. Here, what we implemented was a factor mixture model approach. I'll show you what that looks like in just a second. In our R01, we're doing something slightly different and looking at kind of Mahalanobis distance, using all of this information to characterize kind of distance from a centroid with regard to kind of risk. And but in this approach, we use this factor mixture modeling to look at these profiles. We can then look how these profiles stratify based on the input features. But more importantly, we can verify whether they have some sort of prognostic utility. Do these groups, can they be differentiated on something separate, on a feature that was not included in the modeling? In the R01, at this stage, when we start to phenotype, we also are getting imaging data. So between 24 and 30 months, we're going to get a big set of imaging data from these kids after we select them to be kind of at risk or not. But this whole approach depends on good starting data with parent report. And so this instrument is the VRRSB that I'm talking about, the video reference part. We really like it because you begin by a parent watching a video. It's about a 3 or 4 minute video of a 19-month-old doing great behaviors, great reciprocal social behaviors. And then the first set of items, that little child is the anchor. So you're rating your child against what you just watched. And the hope is that that is going to provide more reliable responses from parents. And we're accumulating a lot of data, kind of validating these measures, but just to give you an idea, it was designed as a toddler version of the SRS. Here's what the shape of the distribution of the SRS looks like in 22,000 school-aged children. Of note here, what you see is a greater proportion of males have higher scores, okay? And that's a very regular finding in kind of among autistic traits. And so, and we have a sample of 1,700 18 to 24-month-olds, and we showed the same pattern and also a pretty good distribution of responses. So just as a starting point, I was really happy to see this dimensionality in our scores. Other measure that we've been working to validate. So this paper, we have three papers or 4 papers on this right now, kind of all telling mainly validation of the instrument, trying to build that story. But the other measure, the repetitive behavior scales for early childhood, we're also got a number of papers out on it, again, trying to validate its utility. When used in combination, what we're going to do is use each of these measures along with the CDI, and we generate these 7 features. So these 4 come from the repetitive behavior scale. This one comes from the VRRSB, the reciprocal social behavior scale. And then these 2 words produced and total gestures come from the MCDI. And the factor mixture model, the idea is that you can identify these latent dimensions, and then in the same kind of model, use those latent dimensions, or in this case, factors, to quantify, to do clustering, essentially, or latent profile analysis. So here we're identifying these 2 factors, one that's atypical behavior, one that's social communication. They're not correlated with one another. And then we use these to derive these clusters or profiles. And in our case, it was a 5-class solution that fit the data best. And so we looked at kind of internal validation of these profile classifications. This is a busy slide, but let me just highlight a couple of things. So our risk groups are going to be, and we don't know who these kids are, but we kind of ad hoc assign a label. that what looks like risk to me are classes 2 and 4. So here's 2, they're both the darkest, okay? They're showing the most repetitive motor behavior. They're showing the most self-directed or self-injurious behavior. They're showing the most rigid adherence to routines, and they're showing the most repetitive play or restricted interests. And these 2 are really interesting, that you get complete separation, stratification of these groups in these measures. This one to slightly less extent. But then obviously these 2 groups are going to be group 3 and 5 are low risk to a large extent. Now, interestingly enough, group 2 and 4, remember, our risk groups. They're also the highest. Here, high is not good. So if you have a higher score, that means more social impairment. This is our reciprocal social behavior measure. Again, so they're where we would expect. But then with regard to communication directly, so gestures and words produced, group 2, which is a high risk group, was producing the fewest words and producing the fewest gestures. But group 4 was doing really well. They were producing the most words and the most gestures. So this highlights this group. It's this very small group that would be missed in typical screening environments context, because their linguistic ability would overshadow all of these other behaviors that are potentially problematic. So with regard to kind of an external validation of these profiles, we brought a bunch of these kids back, over 100 of them. And so yeah, this sample is 1,500 kids. In the risk group, this class was made up of 4 kids. And interestingly enough, it was in our sensitivity analyses, they continued to identify these 4 kids as being this unusual profile. And this sample was like 18. So it's a small group of kids that we're calling at risk out of 1,500. Brought a bunch back, and here's the 5 groups again on the ITSE. So we're extracting these measures of externalizing, internalizing, dysregulation. I know I'm coming down to the wire here. But more importantly, when we combine the kind of high-risk classes, we get these pretty whopping effect size differences and whopping effect sizes, clinically meaningful effect size differences in these high-risk groups compared to the moderate and low-risk group. So here, parents will provide more information if incentivized. We're using this kind of data-driven approach based on multiple relevant dimensions to classify these multiple risk profiles. We have preliminary evidence that the profiles identified have prognostic value. And we think of this as having kind of potential to inform more personalized clinical recommendations, so that, a parent might complete these before they go to their well-child checkup. Their scores get pushed to us in the cloud and computed, and we push back a risk score or risk profile to the pediatrician that they have and are able to kind of personalize a clinical recommendation when the family comes in for the well-child checkup based on that profile characterization. That's the kind of vision here. But in sum, so what we're trying to do, we're trying to innovate with regard to behavioral measurement. I think one aspect that gets lost among neuroimaging folks, I think, is measuring behavior is really hard too, and it takes people, putting their heads together and really trying to do this well. And so we're trying to work on this. I didn't have time to talk about any of my eye tracking work, but this is another domain that my lab is very invested in and thinking about. We've developed this tablet-based eye tracking system. We do eye tracking in the lab very well, but we've now developed this tablet-based eye tracking system that we can take out into communities. We're collecting data in Tajikistan, and we collected data from about 200 kids during a well-child checkup in a clinic in Minneapolis. And so this is something that we're trying to think about. We're also trying to think about carefully quantifying connectivity, and thinking about all the potential sources of artifact that can come from resting state functional connectivity scans, but we're trying to be careful there. Fortunate to have some funding from the Gates Foundation to be working with some of the best people in the world on these problems. One big picture is that we're moving kind of beyond the lesion model, thinking about how multiple networks and network architecture communicates to support complex behavior. And I think this is, the last bullet point is this is kind of our lab motto or mission, is that we're combining kind of this discovery science with public health impact, hopefully trying to, it's not every day do I feel like we're doing that, but that's the goal. So a number of acknowledgements here. for all the grant funding. And then I didn't actually find the grant number for the Gates Foundation grant, but that's been really nice to work with that team of experts on FCMRI. This is my lab from when people could gather, I think last summer. A number of people contributed this data. In particular, I'd like to highlight the work of Suyun Sung, who's fantastic. Colleen Doyle was leading the phenoscreening work. Issa Stallworthy wrote the joint attention and autism paper. Carolyn and Robin have both written several papers on these instruments as well. So I didn't leave a ton of time for question. I want to blame it on Lynn for getting started late, but I should take responsibility. So thanks. I really appreciate it. You guys listening in. I'm happy to answer questions.
Thank you, Jed. I think we'll just thank you for your time and everything that you shared. And if people need to drop off, completely understandable. If you want to send some questions, we can share Jed's email, throw that into the chat, and we can also, you can let me know, but we'll stay on for a few minutes for anybody who has questions and comments to ask. Anyone? Wow, okay. Not even from Dorrit about imaging.
I'm going to make a question. It might sound a little dumb because I'm from the animal world. So I'm very interested about the imaging in small infants. And so I have two questions, one about imaging and one about the questionnaire that you give to the parents. And so the first about the imaging is, do you have a special coil to be able to get that SNR from the babies?
Good question. We're using the 32 channel, the Siemens 32 channel head coil. I have a pediatric head coil at my imaging facility, and I just haven't had the bandwidth to actually do some piloting with it. But now that the Baby Connectome Project, I think, is done, I'm going to get back to it, and we're going to start working with that. One aspect that we wanted to maintain compatibility with the other Connectome projects, and so we didn't want to implement age-specific head coils because it's hard to know what the difference in hardware will introduce versus the actual signal-to-noise changes. So that was a deliberate decision when we were designing the study, that we wanted to use the same hardware, same sequences across the ages so that we can model longitudinal development without having to deal with those unknowns. So there are certainly improvements that could be made with regard to signal noise with an age-specific head coil. Absolutely. Yeah.
And the second question, that's probably the weird one, is when you ask a parent to describe their own kid, and I have no idea about this because it's completely out of my work and my life, but are they like more, I don't know, benevolent about their own children, or then like regular teachers or other people that interact with them? Is there a skewness in the results?
Yeah, for certain types of modalities, absolutely. So you notice that we use words produced, right? When you ask parents to respond or to rate how many words do their kids understand, you get this like striking relationship between maternal or paternal education and words understood, right? Because we want our kids to understand everything that we're saying, right? We think our kids are smart. And so that's why we don't use words understood. Words produced is a little bit, it has a bit more reliability. Now, you do run that risk, and it's not foolproof. Parent report questionnaires, that's why we wouldn't base, for all of our kind of, it's really trying to refine those for the kind of high throughput initial screening phase. There are advantages though to parent report, because parents are with their kids all the time and they get to see their kids across multiple contexts. And there are some advantages there that we can't get when we bring a kid into the lab and we ask them to perform. We have an hour with them and we want them to perform at the highest level in this weird context with weird people. You know, there are limitations there as well. And so ultimately, you know, the best approach is trying to get multiple modalities of information about a child's development, right? But being aware of the caveats is really important.
Have you ever included measures about parent personality or something like that it'd be hard to look at what might bias, if there would be a way to predict bias in these
Yeah,
samples?
we're just getting interested in that. We're going to have some of that data in our new R01, in this phenoscreening R01, where we're getting some data from parents about themselves. When you're asking about language, you kind of don't need it. All you need is maternal education, like how far advanced are they in education. But for other things, it may play a role, and we hope to be able to understand some of that. That's a limitation. My program research, for a long time, I didn't care about parents at all, and didn't just wanted to study the kids. But now we're gradually coming around where we actually need to do some phenotyping with parents as well.
Now that you're a parent.
Yeah.
Anybody else have questions? One of my questions for you, Jed, and part of why I've really appreciated collaborating with you, is a lot of us are thinking about, because we're dealing with a rare population and we can't get large samples, you know, immediately. So we have to do testing over time, and we're trying to accrue enough of a large sample to come up with reasonable, powerful results. And technology changes. And how many times have we been in a position like you described with DTI, where you're trying to guess what's going to be the next best thing and how to relate it. And I just wondered if you had any thoughts about that, you know, knowing that people are starting some new labs. And
yeah, so I think that there's this really exciting movement called integrative data analysis. And one of the major people in this area is a guy named Patrick Curran at the University of North Carolina in Chapel Hill. And what they're trying to do is create analytic pipelines and strategies so that you can collapse across different measures and find commonalities in items, across age, across cohort, across measure, so that you can, you know, if you had one measure on a big cohort and then another measure on another cohort, as long as you have some common data elements that cross the two, you can implement some of these methods to try to synchronize your measurement. It's all kind of the bigger issue is measurement invariance and how we're testing that and how we characterize that. And they've generated a number of very interesting methods and analytic strategies to augment or help with our characterization of measurement and variance. Imaging wise, the corollary is groups working on harmonization across sequences, across protocols, across, you know, it starts with the same hardware, the same sequences and two different sites and treating that they're the same. We've been doing imaging across TIM trios with the IBIS study. It was using a 12-channel head coil, TIM trios, 4 sites. We had human phantoms flying around twice a year for 10 years. We still, you know, you still control for site. There are differences in the TIM trios if you have the same sequences. And so now it's kind of like, okay, well, what about across scanner? What about if you're using a GE, you know, or Philips or Siemens, and you're trying to match sequences? So there's a big effort right now with regard to harmonization across imaging data. It's way beyond my expertise, but I'm kind of, you know, I just see that work happening.
do you work at all with people that are doing fetal imaging?
Not directly. I had been communicating with Mariah Thomason for some time, and I was part of a group planning for this a baby version of the ABCD study, and there were talks, a lot of conversations about fetal imaging and what could be done, and I think that I think it's a really exciting area. I think that there's a lot more work that needs to be done with regard to kind of resting state data. That's her domain and where she's kind of been publishing. But I think a stronger argument could be made. I think that we're further along with being able to characterize aspects of, get T1s and T2s and characterize some aspects of volumetric development, but less so. Sorry, the quick answer is less so. I'm working less with people doing fetal imaging. I'm trying to keep up a little bit on that literature.
Just curious, because Gregor's on.
Yeah.
It's more his world.
Yeah.
Yeah, I would have a question. So as a neuroradiologist, I would be also interested in how you deal with, you mentioned breathing movements, but there are various types of movements in babies when you scan them.
Yeah.
Generally, I guess you're not using any kind of sedation on those at all. And considering resting state and everything, your protocol must take about half an hour or so. So what is your kind of dropout and of cases?
Yeah, dropouts, that's a really good question. I actually am going to have results at the end of the summer on our a success rate paper. But essentially, we do pretty well with, if we get a T1, we're going to get a T2. We're at like 98% there. Overall, all of our scans, the data that I had from a year ago, we were about 87% success rate. A large part of that is that we don't have constraints at our imaging center. So we can wait for babies to fall asleep. If they wake up, we can wait for them to go back asleep. So part of what your question was about sleep duration and success rate, that's going to vary from center to center because of constraints. And so I think we have an unusually high success rate. And we define success very specifically too. We've defined it as getting a T1 and T2 and 10 minutes of resting state. And then if the baby and the baby continues to sleep, then we'll get 10 minutes of diffusion, and then 10 more minutes of resting state, and then 10 more minutes of diffusion. That's the approach that we take. And the rest is this kind of gravy. If we get it, fantastic. But if we don't, we don't. Your other question about motion is we do think about it a lot. I just got money to buy these really fancy headphones and from this company that's making these MR safe, MR compatible noise canceling headphones. I'm sure some people here have seen those. But we bought one pair and got it implemented on our 7T as we were just going to test it first there. But we're going to buy a couple more for the 3T scanners. So that'll mix it up a little bit. For babies under 9 months, we have some custom headphones that have good coverage to seal the ears and to attenuate sound. And then, but they serve two purposes, because then we lodge foam in to make sure that the seal is good. So we're not only attenuating sound, but we're stabilizing the head in the head coil. So we actually do a pretty good job stabilizing the head. And that'll be a little bit, but the problem is that when babies above like 10, 11, 12 months, their heads are too big for these headphones. So then we have to get creative. And it's more of the same idea, but we don't have the kind of seal that we have with those headphones. And hopefully this new headphone product will work really well. And to not only do the seal for sound attenuation, but then we are doing lots of foam and to kind of stabilize the head.
Filippo and Gregor, do you guys, do you do unsedated scans on your kids?
Yes, we do mostly the term equivalent preemies are generally done what we consider as feed and wrap. We actually, I sometimes wonder what those babies are really, well, we do monitor them with pulse oximetry during the scan, but I do sometimes wonder what those babies are really doing inside there. Are they really sleeping or not? Some scanners are equipped with cameras where you can see kind of the eyes of the baby and see if the baby is really sleeping. But maybe that would be another question if you check for that as well.
Yeah, no, I mean, we're pretty sure that our babies are sleeping. It would be the very young babies that you wouldn't know. And I have less experience with that, with babies, you know, less than a month old. The earliest that we've scanned is 1 to 2 month olds. And that would be the cohort that there may be some uncertainty, maybe 2 month old, maybe 2 to 3 month olds too. But over that, you know, if they're awake, they don't like being in the head coil. And so,
Good point.
so we know if they're awake. But we always have we have a baby monitor and someone running the scanner, the console. And so our baby monitor is on a stool, essentially reaching into the bore with their hand on the baby's tummy. And so if the baby wakes up, we get them out and then we swaddle them again and try to get it again. So yeah, it's all during we try to do it all during natural sleep. The one caveat is that, you know, we don't know, and maybe there will be some improvements in this over the next couple of years, but we don't know sleep stage, and that affects breathing rate. That could affect resting state.
Resting state, yeah.
You know, if you're getting into REM, we don't know. And the best way to do that, I've talked to people about it. The best way would be simultaneous EEG, but you know, that kind of setup with babies is really tricky. And I do have a friend at Columbia, Bill Pfeiffer, who thinks that we should be able to do it without EEG. So that's something for the future. That would be a big bummer if we show that resting state varies by sleep stage in these kids, because it would be, we'd have all of this data where no one knows the sleep stage, and it would be this like, I don't know. So.
Okay. Any other questions? We should probably wrap up. Thank you, Jed.
Yeah. Thanks for the opportunity. I really appreciate it, and look forward to gathering with all of you when we can in the future.
Yeah. All right. Take care, everybody. Thank you for being on.
Okay. See ya.
Bye-bye.
Right.